All right, here we are with video 2 of our dihybrid series. And I think I should be able to finish this all in a second video. So this hopefully is video 2 of 2. In our first video, we started talking about what is a dihybrid cross, how to use foil to find the gametes, how to do a big 4x4 Punnett square. And then I said right at the end, what if you have two parents that are not heterozygous times heterozygous? So here's the example that we're looking at here. We have heterozygous for height and heterozygous for green seeds, but the second parent is not heterozygous. That second parent is recessive. So we're going to use the same letters here that we did for the alleles in the previous example. So for height, we have big T for tall, that's our dominant. And it is dominant to the little t, that's the recessive allele. And again, we use the same letter for each trait. So big T for tall, not an S for short, but a little t for short. And we will keep our letters together. Then we're going to look at the color of the pea seeds. That is our second trait. Big G for green is dominant to the little g, and that is the yellow, the recessive allele. So our first parent, heterozygous tall, so it's heterozygous for height, and heterozygous for green is going to be big T, little t, big G, little g. And the other parent is homozygous short, homozygous yellow. So what do we do? Well, we're going to use FOIL again, and we're going to determine the gametes from each parent. So the first letter in each pair, so here's a pair of T's, big T. Then the first letter in the G pairings is the big G. So that's how we got our first gamete, big T, big G. And then the next step, the next thing we're going to do is look at the outside letters of each pairing. So the outside letters, there's our big T and a little g. That's how we got that gamete. Next up, our FOIL tells us we're going for the inside letter of each pair. So the little t and the big G. Little t, big G. And the last step here, well, last, L for last letter of each pair. And I'm doing those in black. So the little t and the little g. And there's our double recessive. Now, do we have to use FOIL for the second parent? No, we don't. All that this parent has is little t's and little g's. So we have the double recessive. If you want to write them out, you sure can. Have at her, right? Go right ahead. Little t, little g, little t, little g. Do it 4 times over and do a 4x4 Punnett square. You can just do it just to prove my point, just to show that you're going to get the exact same ratios, right? Here we have a big T and a big G. So that means that every single one here is going to have a big T and a big G. It's going to be tall and green. Doesn't matter what that second allele is. Big T, big G, tall and green. Same for this one. Big T, big G, tall and green. So if you want to do a big 4x4, you're going to see that 4 out of 16 are tall and green. But is that lowest terms? No, it's not. It's 1 out of 4. So you can reduce that to 1 out of 4. So why would you do the big 4x4 Punnett square when you can just do the shortcut, right? 1 out of 4 is tall and green. 1 out of 4 is tall and yellow. 1 out of 4 is short and green. 1 out of 4 is short and yellow. So 25%, 1 out of 4, or 0.25 for each one. No point in you wasting your time doing the 4x4. If you want to do it just to see that the ratios are the same, you can, but by no means do you need to. Okay, and so that was our shortcut method, the 4x1 instead of 4x4. The whole point, it's all about the ratios, and so you're getting the exact same ratios. So now you can read through some more examples and do a couple of questions practicing. Here's my example to you where we are looking at poultry. So poultry, those are chickens, turkeys, okay? And we have poultry, it says crested head is our dominant allele and the plain head is the recessive. So I've decided when I'm in a hurry and I'm doing this, my big C's and my little C's are going to look a lot alike. And so I've decided that I'm going to use the handwriting on my little C, and my handwriting, and so it kind of has a tail on it a little bit. So the little c here is my recessive. Okay, and then what is the other trait? This is a dihybrid. The second trait is black feathers, dominant to red feathers. That's our recessive. Okay, so what's my next step? Well, my next step is let's set up what the parents are. I'm going to read through what it says. It says the first parent is homozygous for black feathers, and this word homozygous also refers to plain head. Okay, so homozygous black, homozygous plain. The second parent, also homozygous, but this one is red and crested. So I got my phenotypes, the physical appearances. Are they black or red? Are they plain or crested? Now I'm going to set up my genotypes. So homozygous for black, 2 capital letters, big B, big B. Homozygous for plain, little c, homozygous red, homozygous crested. Okay, what's my next step? Well, my next step is gametes. So I don't actually need to do FOIL here. I can see that there's only a big B and a little c from this parent. There's a little b and a big C from this parent. If you want, you can do all 4. Go right ahead. I'm not stopping you, but, well, actually I am. Why would you waste your time doing that? Don't do the repeats. Just do a 1-by-1 Punnett square. Instead of a 4-by-4 where you get 16 identical offspring, just do a 1-by-1. So here's our 1-by-1 Punnett square, and we have a big B. So all of the offspring are going to have black heads, and then big C, little c, well, they're all going to have crested heads. Okay, so here's a crested head. Looks like he's having a bad hair day, but every one of these offspring in our F1 generation is heterozygous. Okay, so black feathers, crested head, they are all heterozygous for this F1 generation. Now what? Well, what does the question say? Let's see. It says if the F1 are allowed to mate, so we're going to go F1 times F1, what do we get in the F2? Any bets on it'll be a 9 to 3 to 3 to 1? I think that's a pretty safe bet, right? We've got a heterozygous times a heterozygous. So you are correct in saying that when you FOIL the gametes out, you're going to get all 4 different gametes. And then you end up with a 4-by-4 Punnett square. Okay, so first, big B, big C. Next up. Oh, outside, big B, little c, big B, little c. Okay, next what? Inside, little b, big C, little b, big C. What's the last pair, letter in each pair? Little b, little c. Okay, so there's our 4 gametes. There's no repeats here, right? It's not like we have big B, big C, right? We're not repeating 2 of the same. There's no repeats. There's all 4 different ones. And you know that there's going to be 4 different gametes here as well. So you are going to set up a big 4-by-4 Punnett square. And when you do that, it will look like 4 gametes, 4 gametes. There's our 4-by-4 Punnett square. Okay, so a couple of tricks of the trade. This first column going down has a big B and a big C. That means all of the offspring are going to have big B, big C, each and every single one, right? They're all going to be black and crested, black and crested. Doesn't matter what that second allele is, they're all black and crested. Likewise, going across this way, all black and crested. Which other ones are going to be black and crested? Big B, big C. So this guy, black and crested. Which other ones? Black and crested, black and crested. Also going to be this guy here. So what are we getting? Those are the 9 black and crested, the 9 double dominants. Okay, then what? Well, then we are going to have big B, something, little c, little c. Big B, something, little c, little c. And one more where we have a big B, something, little c, little c. So there is our 3 that are black, big B, something, little c, little c. What's the little c stand for? Well, that is our recessive, and the little c is going to be the plain heads. Okay, not done yet, almost there though. let's do the next set. Okay, and again, I like color coding these. Well, here we go. Little b, big C, something. Another little b, big C, something. I'm gonna make that look better like a big C. And we have one more. Here we go. Little b, big C, something. So there's our next three. Little b, for they are all red feathers, but they have that dominant big C, and so they're going to have crested heads. Doesn't matter. Again, it could be big C, sorry, or big C, little c. So I'm just going to put a blank there. Now, who's left over? Well, you guessed it. Here is our double recessive. Little b, little c, little c. 1 out of 16 is going to have red feathers and is going to have that plain recessive head, not the crazy bad hair day. So 9 to 3 to 3 to 1 is our ratio. And again, we expected that, right? That was a Mendelian ratio. We will always get that when we have a heterozygous individual times a heterozygous. So here's just my nice looking key of that, 9 to 3 to 3 to 1 ratio. Okay, so you got some practice to do now. Get good at doing these dihybrid crosses, 2 trait crosses. And again, if you have any questions, just let me know. Michelle.Ledeen@ghsd75.ca. Till then, take care.